Hi everyone and welcome to Brittany Vlogs. I have no video editing expertise at all, so I'm just gonna talk at you for like 15 minutes. I'm really sorry. But hopefully you find the information interesting, at least as interesting as I do. So let's start. I'm gonna start off by talking about the debate of free will and neuroscience for something that we learned in class that I found really interesting. I personally find this topic really interesting because it combines so many different disciplines like philosophy and neuroscience into this debate. I love many different areas of study like biology, philosophy, psychology, which is why I'm majoring in psychology and why I'm so interested in neuroscience in general. The study that set this debate into motion was a 1985 experiment by Benjamin Libet, or Libet. I think Libet is better, but I guess it's pronounced Libet. In this experiment, Libet asked participants whose brain activity was monitored using an EEG to flex their wrists whenever they had the urge or desire to move. At the same time, they were instructed to keep track of a spot of light that was like moving in a circle, and they were to note the position of the spot when they first had the urge or desire to move. This was called W time. The EEG was used to find the readiness potential, which is a measure of activity in the supplementary motor area of the brain that leads up to voluntary motor action. Libet found that the onset of the readiness potential occurred up to 550 milliseconds before the participants reported their W time. Libet interpreted this as evidence against free will, as motor preparation began before the agent had an urge to move, or reported an urge to move. But more recent experiments have presented more evidence in the field of neuroscience and the debate against free will. Some studies I find particularly interesting are the Soon et al. Studies, because they utilized an fMRI instead of an EEG. In 2008, Soon and colleagues asked participants to press either a left or a right button whenever they felt the urge to do so. fMRI scans revealed that activity occurred in the prefrontal and parietal cortices up to 10 seconds before the participants reported their desire to move. And in a second study done in 2014 with a very similar experimental design, Soon and colleagues examined the brain activity while participants were asked to make more abstract decisions that involved adding and subtracting numbers, and they found similar results. So a study that I find that is like crazy interesting is the 2009 Desmarget study, in which participants were undergoing awake brain surgery to remove tumors. They found that when they stimulated the parietal lobe at low intensity, participants reported that they felt a desire to move their hand or lick their lips, and when they stimulated at a higher intensity, they actually believed that they had done so, though no movement actually occurred. However, when they stimulated the motor cortex, at low intensities, slight movement of fingers or the lips occurred, and at higher intensities, a whole limb would move. However, the participants actually had no recollection of this happening and even refused to believe the doctors when they told them that they had in fact moved. This shows that feelings of volition and agency are not necessary for movement, only awareness that movement has occurred. And they even can give the illusion of movement when it hasn't actually happened. And furthermore, this could suggest that intention and conscious awareness are not actually precursors for voluntary movement, but could even be a consequence to the movement itself. I could talk forever about this topic, but I'm going to move on. So I'm going to end with some research that's done by faculty here at Chapman. Dr. Uri Maus at the Brain Institute conducted a study in 2019 that looked at readiness potential onset for arbitrary versus deliberate decisions. So for deliberate decisions, these are decisions with like intention or purpose. In the study he did, it was like you like were assigning money to like you would like choose between different like charity organizations. So like one of them was like for like anti-abortion and one of them was for like gun control or something like that, versus like arbitrary decisions which like don't have any meaning behind them. Interestingly enough, he found that when deliberate decisions were made, there was actually no readiness potential that was present, whereas there was a consistent readiness potential for all arbitrary decisions, which could have some interesting implications towards the actual role of the readiness potential in the brain. I'm going to move on to my second thing that I found super interesting in our class, which is addiction and substance abuse. I think substance abuse is a really important topic to study because it impacts all of our lives, or it will at some point or another, whether it's us that will be using or someone close to us. I could probably talk forever about the different like biological mechanisms behind addiction, like the mesolimbic dopamine pathway or the ventral tegmental area and the nucleus accumbens and the role of opioid receptors and whatever else. But instead, I want to talk about the different theories and models of addiction and why the way we talk about addiction is really important. Most of this information is from a TED Talk by Johann Harris, which I'm going to link below, and I encourage all of you to watch it. It's a really, really good TED Talk. So one of the first theories is the gateway theory, which I'm sure we're all familiar with. You know, you try alcohol or nicotine, or what DARE tends to emphasize, marijuana, and then you get addicted to more dangerous drugs as a result because you're introduced to them. There is some truth to this theory. Obviously, meth users don't just start with meth. They're going to start with alcohol or weed or something and then move on to harder substances. But this theory makes one of like the biggest mistakes known to science and research. Correlation does not equal causation. It doesn't account at all for the fact that the majority of marijuana, alcohol, and nicotine users never go on to use harder substances. And it also kind of pushes aside the fact that nicotine and alcohol are also extremely addictive substances. It's not just meth, it's not just crack. Alcohol is very dangerous and has a huge potential for addiction. The medical model of addiction, which views addiction as a disease based on evidence of dysregulation of brain function, and it underlies most professional treatment like the 12-step program. It's good for a lot of reasons, like insurance coverage for therapy and medication. There is less self-criticism or stigma associated with it. You're less likely to punish harshly based on it if you're not viewing it as like a moral issue and instead as like a medical issue. And it accounts for brain-related changes that occur over time, and then also factors like genetics and epigenetics. But you can't test for it neurologically. It has reduced self-efficacy associated with it, and it doesn't account for those psychosocial factors or people who can overcome addiction without formal treatment, or people who are just prescribed like opioids for pain, or who are given like intravenous morphine at the hospital and don't end up addicted to heroin. So I think we need to start addressing substance abuse as a psychosocial issue, and here's why. I think we're all familiar with the rat experiments in Rat Park, but just in case, I'm going to summarize it. So basically, a rat was placed in like an empty cage, and they're given 2 bottles of water. One is just plain water, and then the other one has cocaine in it, and then the rat will repeatedly go back to the water with the cocaine. But that's under experimental conditions where the rat's just in an empty cage with these 2 bottles of water and then like food. But if we put these rats into what was called Rat Park, which was basically like rat heaven, the rats had like tons of toys and treats and like lots of rats to like play with, they wouldn't even touch the cocaine water. Like it didn't matter to them at all. They would just drink the normal water. And so there actually is like a human version of this experiment, which is the Vietnam War. In Vietnam, 20% of American troops used an excessive amount of heroin while in Vietnam. But when they returned home, 95% of users just completely stopped without any withdrawal symptoms or without the need for rehab. So this shows that addiction isn't necessarily an issue based on biology or chemical hooks that we're usually taught. It's actually about environmental adaptations. Some experts have actually suggested that addiction should be renamed bonding because humans have an innate need to bond and connect with each other, but when that need is interrupted by trauma or isolation or just the unpredictability of life, people will bond with other things like an unhealthy relationship or gambling or drugs. Most of us don't compulsively use drugs because we don't want it to disrupt our life. We love our friends, we love our family, we care about school, we care about our jobs, we have things that give us support and value. But when we don't have these connections, we turn to other things for that support and value. And this is why the war on drugs is inherently flawed. When we criminalize drugs, we put people in prison, we make it harder for these people to get jobs or seek education. Basically, we further take away the things that are shown to prevent drug use. And if you need any further evidence that substance abuse is a social problem and decriminalization actually does work, in 2000, Portugal was one of the countries with the worst heroin addiction rates in the world. After they decriminalized all drugs and put the money that would be used in the prison systems for drug addicts into instead job rehabilitation programs, the rate of injectable drug use dropped by 50%. So that's why I think I'm always an advocate for the decriminalization of drugs and drug reform. So now I'm going to talk about some things I wish we talked about in class a little bit more. First, I'm going to start with psychedelics. We covered it a little bit in class, but I'm going to talk about it more just because I think it's really interesting. I find psychedelics interesting for a lot of reasons, not just because they're like cool or mysterious, but they pose a potential treatment option for lots of different mental health issues. MDMA is getting lots of traction in relationship therapy, and LSD and shrooms are being shown to treat depression, anxiety, PTSD. But today I'm going to specifically focus on shrooms or psilocybin for the treatment of substance abuse disorder. Psilocybin is a prodrug that is dephosphorylated into psilocin, which actually produces the psychotropic effect that's associated with the drug. The molecule structure is very, very similar to serotonin, which gives it a high binding affinity to 5-HT receptors, and it makes it a serotonin agonist or partial agonist. It has a high binding affinity for most, if not all, 5-HT receptors, but it probably has the highest affinity for 5-HT2A. The cerebral cortex contains high densities of 5-HT2A receptors in all four lobes. 5-HT is thought to have function in complicated information storage and manipulation, self-control, and flexible and adaptive behavior, and the use of psilocybin only enhances these traits, which is very beneficial if you're looking to end an addiction. As for specific mechanisms of the drug, an fMRI study found that psilocybin significantly decreased cerebral blood flow and bold signal in many brain regions, but most notably the medial prefrontal cortex and the postcingulate gyrus, decreasing the interaction between the two regions. This can be explained by evidence supporting that the activation of 5-HT2A receptors will increase GABAergic transmission, along with pyramidal cell inhibition, leading to an inhibitory response. The PCC is associated with the default mode network, or the DMN, which contains the highest number of cortico-cortical connections in the brain, and thus allows for an efficient transfer of information by using the least amount of neuronal connections as possible. The DMN is central to day-to-day functioning and integration, and has also been associated with self-referencing. The decreased activity of these neural networks is hypothesized to be the main factor of the properties of psychedelic drugs like psilocybin, as it allows for loose and unrestricted cognition. So despite this evidence for the mechanisms of psilocybin and other psychedelics, the direct mechanism that produces the hallucinogenic and profound effects, along with its ability to treat substance abuse disorders, remains mostly unknown, actually. Despite the profound effects on the disorder, the two pathways that are central to drug reinforcing that will create addiction, the mesolimbic and the mesocortical dopamine pathways, are not implicated to be affected by psilocybin at all. And so as a result, the specific mechanism behind its treatment of substance disorders remains a complete mystery. But the potential for use of psilocybin for the treatment of substance abuse disorders is only beginning. The results of many studies have been groundbreaking for the treatment of this disorder. A study that included participants who smoked at least 10 cigarettes a day and had a history of unsuccessful attempts to quit ran for 15 weeks long. Sorry, that was gross. They had psilocybin sessions at weeks 5, 7, and 13. Following the study, 80% of participants had abstained from smoking at the 6-month follow-up, and 67% of participants remained abstinent at the 12-month follow-up. Another study investigated psilocybin's effects on alcoholism. It found that subjects diagnosed with alcohol dependence reported decreases in craving and withdrawal symptoms up to 36 weeks post-psilocybin administration after only 1 to 2 sessions. So clearly, psilocybin has excellent efficacy for treating these disorders, and we're only beginning to see the potential of this revolutionary drug. I'm sure you all know, Oregon made history by becoming the first state in the United States to legalize psilocybin. You can only access it at treatment centers if you meet the qualifications for it. So lastly, the last thing I want to cover that we talked about a little bit in class, but I just find really interesting and I wanted to talk about it more, is sensory systems. I think senses are super cool because it's what we use to experience our world. Everything we know about our world, everything we know about reality is just through our senses. So let's talk about it. We covered the auditory system in class, but I want to talk about my favorite and, in my opinion, the coolest sense, which is taste. Your tongue has about 10,000 different taste buds, which act as your taste sensory receptors. They're specialized epithelial tissue, so they synapse to nerves when sensory transduction occurs. When certain chemicals in our food, or tastants, dissolve in your saliva, they're able to bind to the receptors on your tongue. We have 5 main taste sensations: sweet, salty, bitter, sour, and umami. A quick myth to dispute: I'm sure everyone has seen those maps of the tongue that are like, oh, like salty is like at the back and like bitter is like on the sides or whatever. That's not true. All tastes are pretty much registered in the same parts of your tongue. It's like all equal. So I'm going to talk about the specific mechanisms that allow you to taste each of these 5 flavors or sensations. So for salty, this is actually just positively charged sodium ions. They just flow through those ungated sodium channels, and they're going to cause graded potentials, which will trigger an action potential. For sour, you're actually tasting acid. The acid will give up a hydrogen ion, which flows through a hydrogen channel and will cause a graded potential, which again in turn triggers an action potential. Sweet is glucose, as you probably guessed. It acts as a ligand and binds to metabotropic receptors, which will then open cation channels and again trigger an action potential. Side note, artificial sweeteners are glucose agonists. I was like, when I first heard that, I was like, oh, that makes so much sense. Like, I don't know why I never thought of that before. Like, obviously, but I just think that's kind of cool. So for bitter, you're tasting a base. The base will act as a ligand that binds to metabotropic receptors, which again opens a channel and triggers an action potential. And then this is the coolest one in my opinion. Umami, you're tasting glutamate. You're tasting a neurotransmitter that's like actually in your body. Glutamate acts as a ligand and binds to metabotropic L-glutamate receptors, which causes an action potential. And MSG, which is like what they put on like McDonald's hamburgers or whatever, is actually monosodium glutamate. So if you've ever wondered what glutamate tastes like, it tastes like McDonald's hamburger. Not actually, but kind of. And then just like a fun little fact at the end, miracle berry, which are like those fun little tabs that you can put on your tongue and makes everything sour taste sweet. It's actually a glycoprotein that acts as a sweet taste agonist in the presence of sour foods or acids. So that's just like a fun little fact that I thought I would add. I hope you guys enjoyed watching my video. I really enjoyed this class semester and I really enjoyed getting to know you all, and I hope you have an amazing winter break. Thanks, bye.